Unit 3.3, Endosymbiosis. You have a one-page section in your note-taking outlines, page 35, that has a few self-study questions about endosymbiosis, and we will be covering those in this lecture, but it would be easier for you if you followed along in this lecture using the endosymbiosis supplement that I posted in D2L recently. So if you don't have that, you might want to pause here and at least print yourself a copy. So I think that it will be more easy. I think it will be easier for you to follow along. Eukaryotes evolved from their precursors, the prokaryotic cells, and all of this happened about 2 billion years ago in the process called endosymbiosis, the theory that was first brought forth by Dr. Lynn Margulies. If you look at the bar graph on this page, you'll see that about 3.75 billion years ago, prokaryotes appeared, and then about 2 billion years later, the eukaryotes appeared. So eukaryotic cells have only been on this earth for about half the amount of time as their primitive precursors, the prokaryotic cells. Endosymbiosis is derived from a combination of terms, the first being endo, meaning inside, and the second being symbiosis, and that comes from the Greek sym, S-Y-M, meaning together, and bios, to live. So symbiosis is a close association between two organisms, and endosymbiosis is this close association with one organism living inside the other. And I do want to point out that your textbook does interchangeably use the terms intracellular symbiosis and endosymbiosis. And if you think about those two terms, you'll realize that they are actually referring to the same thing. Before we begin our 5-step discussion of the theory of endosymbiosis, I wanted to just give you a little summary of it. The theory of endosymbiosis says that once upon a time there were large prokaryotic cells that had small prokaryotic cells that got trapped inside of them. And you can see in the diagram on this page the image of a large prokaryotic cell and a small prokaryotic cell. So imagine those two sort of side by side existing in the same environment and getting closer and closer together. Once those small prokaryotic cells got trapped inside the larger prokaryotic cells, the small prokaryotes, evolved into organelles. And the eukaryote in the image below the little arrows there joining the small and large prokaryotes is a representation of a cell in which small karyotes over the years have evolved to represent all some of the organelles within that eukaryotic cell. And eventually these small prokaryotes trapped inside develop specialized functions of their own. And that's true today, that each type of organelle has its own specialized function. So in the first slide you saw a large prokaryotic cell getting small and trapped inside. In this step, those small trapped cells began to reproduce inside the larger cells. Next, the trapped cells began to perform special functions, and the larger cells, the larger cell that had the small cells trapped inside, was able to benefit from those functions. Eventually, these cells that were trapped inside the larger cells evolved into organelles. They lost their ability to carry out all of the functions of life, and they became specialized to just perform one function. For example, we have mitochondria that produce the ATP energy for the cell. We have ribosomes that function to make proteins for the cell. Those are just two examples. And then at last, those larger cells that had the smaller prokaryotic cells trapped inside began to rely on these organelles for metabolism. They were no longer able to carry out their own metabolic functions. They needed those organelles in order to survive. Happening right along with the formation of organelles from small prokaryotic cells was another cell structure evolving. That cell structure was the nucleus. The nucleus evolved not through endosymbiosis, but through a separate process. If you look at this diagram, you'll be able to follow along and see that, as you know, the eukaryotic nucleus is DNA wrapped up in the nuclear membrane. And what happened was a large prokaryotic cell formed a membrane using its own cytoplasmic membrane around its own DNA. So it was a prokaryotic cell membrane that formed around its own DNA to make the original or the precursor to the nucleus. Please take a look at your self-study section on page 35 of your note-taking outline and think for a minute about the answer to these two questions, and hopefully when you were filling out your self-study you have some notes for yourself. Which prokaryotes, the archaea or the bacteria, were the most likely precursors to eukaryotic cells, and why? And then, which two organelles gave scientists the clues about the origins of eukaryotes? Explain your answer. Archaea were the most likely kind of prokaryote to be the precursor to eukaryotic cells because, remember, archaea do not have a cell wall. So without a cell wall, the flexible membrane of the archaea could more easily reach out and engulf smaller prokaryotes and pull them into the archaea's own cytoplasm. And then for the second question, it's the mitochondria and the chloroplasts that give us clues about the origins of eukaryotes. Because, remember, the mitochondria and the chloroplasts have ribosomes. And those ribosomes are the same size as prokaryotic ribosomes. So just picture a prokaryote being engulfed by an archaea, being drawn into the cytoplasm of the archaea, and then over millions of years, those prokaryotes with their 70S ribosomes began to evolve within the archaea into mitochondria or chloroplasts, taking on the functions of production of energy for what was to eventually become a prokaryotic cell. Here are a few more details about why scientists think that mitochondria give us clues to the origins of eukaryotes. Mitochondrial DNA has a circular chromosome, just like prokaryotes do. And those chromosomes have some of the same DNA sequences of prokaryotes. Mitochondria replicate all by themselves. They don't rely on the cell itself for replication. They divide independently of the cell. And as you do know, mitochondrial ribosomes are the same size as bacterial ribosomes, 70S. As a summary, this is just a reminder that the pre-eukaryotes benefited from those prokaryotic cells that they engulfed. The engulfed prokaryotes that later became mitochondria produced ATP, and the larger pre-eukaryotic cells that eventually became eukaryotes were able to benefit from those mitochondria. They were able to use that ATP that those engulfed prokaryotes produced for them. Engulfed prokaryotes that later became chloroplasts were the cyanobacteria. And you remember the cyanobacteria used the sun's energy to make ATP, and then that ATP could be used by those larger cells that had engulfed those cyanobacteria. And then the cyanobacteria evolved into the chloroplasts. So in a very long leap of time, these single-celled organisms evolved into multicellular organisms, and this is a scientist's theory about how that happened. Eventually, the single-celled organisms started to form little groups. The groups became permanent. They were called colonies. Eventually, these colonies themselves began to group together, and the colonies developed specialized functions. So a colony on the right side of an area maybe had one function. A colony in another section of an area developed another specialized function. So each colony's specialized function allowed those colonies to evolve into tissues and organs. And eventually, these individual cells that form these colonies lost their ability to survive apart from the colonies, and the colonies lost their ability to survive apart from each other. We have reached the end of the last unit. This is Unit 3.3. I hope that you have a great rest of the week. I am interested in your feedback about the recorded lectures, about the handouts that went along with them, and about the activities that I've added to try to help keep you engaged in the course and engaged with each other even though you're not getting to see each other. I look forward to seeing you on Monday, and I look forward to hearing from you either by phone or by email with questions or comments that you have. Thank you very much for your cooperation this week, and I will see you soon.